Hello, guys. Guys, we are going to start our PPT on the topic of 3D bioprinting.
Good afternoon, everyone. Today, me and my teammates Hardik, Janardhan, Shalu, me, myself, Ridhima, and Prasad are gonna present on topic 3D bioprinting. I would like to ask anyone: has anyone ever heard of the term 3D bioprinting? Anyone?
Maybe it is related to print something.
Yeah, like you are partially correct. So now I would like to introduce you to 3D bioprinting. It is an innovative technology that applies the principles of a 3D printing to create complex biological structures such as tissues, organs by layering biomaterials, cells, and growth factors. The ultimate goal of bioprinting is to mimic the architecture and the function of native tissues, offering solutions for medical challenges like organ shortages and personalized medicine. Next slide, please. 3D bioprinting builds on the principles of additive manufacturing but goes beyond conventional 3D printing by using bioinks. A mixture of living cells and supportive materials is rather used than plastic or metal. The bioprinted structures are designed to promote cell viability, organization, and function, simulating real tissues found in human body. The key components are bioink, which contains the living cells that are suspended in a gel-like material. Bioprinter, a specialized 3D printer capable of depositing bioink, blueprint, or design. It is a patient-specific 3D model generated from imaging techniques. Next slide. Talking about the definition of 3D bioprinting, it has evolved from 1980s early 3D bioprinting innovations to a transforming tool in healthcare. In early 2000, researchers successfully modified printers to deposit living cells, and by 2010, companies like Organo demonstrated functional tissues like blood vessels and liver models. Advances throughout the 2010s enabled the printing of complex structures such as cartilage, skin, and bone. By 2020, bioprinting achieved vascularized tissues and organoids for research through challenges in scalability and viability remain. With ongoing improvements in AI, bioinks, and regulatory frameworks, 3D bioprinting holds the potential to revolutionize medicine through personalized treatment, organ fabrication, and improved drug development. Next slide, please. The importance of a 3D bioprinting in bioengineering is tissue engineering, regenerative medicine, personalized medicine, and surgical planning and training. Talking about the tissue engineering and regenerative medicine, it helps in creation of a functional tissue. Bioprinted tissues such as skin, cartilage, and bone can be used for patients with injuries or degenerative conditions. Organ replacement research. Bioprinting aims to solve the global shortage of organ donors by creating functional organs, personalized medicine, patient specific implants, and prosthetics. Bioprinting enables the creation of a prosthetics and implants tailored to the patient's anatomy, improving comfort and functionality. On-demand printing, on-demand printing in surgery. On-demand printing in surgeries, um, time and improving outcomes, surgical planning and training, stimulation models. Bioprinted models of organs and tissues are used for pre surgical planning, helping surgeons practice complex procedures. For medical training, 3D printed anatomical models provide students with hands on experience in a risk free environment. Now,
Thanks,
now
Ridhima.
my, yeah.
Thanks, Ridhima, for having covered the introduction and the historical development of 3D bioprinters. Now, let's deep dive into the various bioprinting techniques, the compositions and the compartments of bioinks, and much more. Now let's take a closer look at the three main approaches that drives this technology. So the first one is the biomimicry, the second one is the autonomous self-assembly, and third one is the mini tissues building blocks. Biomimicry, as the name says, involves replicating the exact architecture and the function of natural tissues or organs. This approach focuses on intimating the extracellular matrix, cellular organizations, and vascular structures by precisely reproducing the biological blueprint of the tissues. The goal is to create an engineered tissue that closely mimics the original both structurally and functionally. The second, autonomously self assembly, takes inspiration from natural embryonic development, where cells organize themselves into complex tissues without external scaffolding. This approach relies on the inherent ability of the cell to proliferate, differentiate, and self organize. The mini tissues building blocks involves assembling small functional tissue units or organoids which are then fused to create larger, more complex structure. Each mini tissues functions as a building block mimicking the specific part of the organ and the tissues. Now here we can see in a short way how this bio 3D printing is basically. Here we can see in this diagram there are polymers which consist of cells, there are cross-linker polymers. At the end, the mixture of this all forms the biolinks, and this can be used through various different technologies, which are the extrusion-based bioprinting, inject-based bioprinting, and laser assistance bioprinting, which leads to the final product in the in vitro disease model. So now, as I has told, that now we will look into the various stages of bioprinting. At its core, the 3D bioprinting is based on the layer by layer deposition of biological material to create complex three-dimensional structures. Unlike the traditional 3D printing, which issues synthetic materials like plastics or metals, 3D bioprinting uses bioinks, a combination of living cell biomaterials and growth factors that are specifically designed to replicate the properties of human tissues. As shown in the previous diagram, or in this diagram you can see biomaterials plus cell gives the ink, plus the mesh diagram which has been faded to the bioprinters. Now let's break down this whole diagram or the whole process into three essential stages. The first is the pre-bioprinting stage. This stage focuses on preparation. First, a digital 3D model of a tissue or organ is created using CAD, that is the computer aided design software. This model serves as a blueprint guiding the bioprinters in a precise placing cells and materials. Nextly, in it we can use the bioinks. The second stage is the bioprinting stage. Here you can see in the middle above the processing, here is a micro extrusion. Here how the machine is placing the cells. In this stage, the bioprinters deposit ink layers by layers by deposit to build the desired 3D structures. Depending on the complexity and the purpose, different bioprinting techniques can be used. Then the last and the third is the post bioprinting stage. Once the 3D structure is printed, it undergoes a maturation phase. During this time, the cell begins to proliferate, differentiate, and organize themselves into functional tissues. Now, as we have seen how the process works, now we will look after the different types of technologies which are present. So there are numerous technologies, but mainly which exist are the extrusion based bioprinting, inject bioprinting, and the laser assisted bioprinting. Now in here are the diagrams of various bioprinting technologies. Now here we will look one after another. Extrusion based bioprinting. The bioprinting is the most common method where bioinks is pushed through a nozzle. Here in the third diagram we can see how it works. In a continuous stream to create 3D structures, this method can handle high viscosity bioinks, making it ideal for printing large complex tissues scaffolds. The advantages are it is suitable for printing tissues that require structural strength such as bone and cartilage. The first one is the inkjet. Now we will look after it. Uses a droplet based approach similar to traditional inkjet printers. It deposits precious droplets to the bioink layer by layer to build tissues. It is known for its speed and cost efficiency. The third and the most important is laser assisted bioprinting. The laser assisted bioprinting uses laser pulses to deposit bioink onto a substrate. The laser energy creates a small focused force that transfers cells and bioinks into high precision without coming into direct contact with the biolink. In the second diagram, we can see how the laser assisted process works. The advantages it offers is the exceptional resolution and the cell viability, making it ideal for printing integrate tissue patterns like vascular networks. Conclusion: each of these technologies has its own strength and limitation, and the choices of method depends upon the various types of tissues being printed. Now, as we have gone through different types of technologies which are present, but the one thing which is common is the bioprinter. So what is a bioprinter? A bioprinter is a specialized 3D printer designed to create biological structure by precisely depositing layers of bioinks which are made from living cells, biomaterials, and other supportive components. The bioprinter translates a digital 3D model into the physical, biological construction, offering incredible precision in tissue engineering. Here, some of the key components of bioprinters can be the print head. As here we can see, there are three exchangeable print heads in which the bioinks can be stored, and through this the printing takes place. The substrate or the build platform and the control system. This control system specifies the CAD model through which we can use. Here is a explained diagram for what the bioprinting is all about. Now we can look, as we have told, there are different types of bioinks which has to be stored. So bioinks are compatible materials composed of living cells. This can be designed to mimic the natural environment of tissues, allowing cells to grow, differentiate, and function efficiently. Now we have looked through how the machine works, different technologies. Now I will call my other friend to explain more.
Thank you, Prasad. Tissue engineering. Tissue engineering is a multidisciplinary field aims to create the biological sustainabilities that is the restore and maintains the improval of the tissue functions. It often involves the use of scaffolds and cells and the biomaterials to generate tissues such as skins and the organs. Key components of the tissue engineering include cells and the biomaterials. First is the cells. The building blocks of the tissues obtained from the various sources such as the stem cells and embryonical stem cells. Biomaterials. Biomaterials, scaffolds materials that provides the structural frameworks for the growth and the development. Next slide, please. Drug testing and the developments. Drug development is the complex process involves the uh, research and clinical testings and the regular approvals. It facilitates the challenges like high failure rates and the long timelines and the high costings. This trends includes the personalized medicines and the biotechnological and the complex complications in the designs and the AI. Next slide, please. Personalized medicines. Personalized medicines is the medical approach that aims to titrate the treatments of the individual patients based on their unique genetical materials and the molecules and the clinical characteristics. This approach recognizes the personalized response different to the um, medications and the treatments. He personalizes the medicines. The genetical testings analyze the genetical materials to identify the risk factors and predicts the treatments. Biomarking identifications identifying the molecules marks that can use to monitor the diseases progressing and the treatments efficiencies. Next slide, please. Technical challenges. Technical challenges includes cell viabilities and the proliferations. The maintaining the cell viabilities during the engineering processes is crucial. The cell must only survive but also proliferates and differentiate appropriately to, to form the further tissues. Mechanical properties. The engineering tissues possess mechanical properties that matches those of the native tissues to further correctly. Vascularities ensures the adequate the blood supplies to the engineering tissues is one of the most significance handlings. Without the facilitating the vascularized networks and the tissue connections survivals. Next slide, please. Environmental factors. The local microenvironments and the includes the facilities such as oxygen testings and the native supplies and the mechanical forces plays the vital roles in the tissue regulations. Next slide, please. Ethical considerations and the regularities. Animal testings, the personalized studies offered the individual animal models, which raises the ethical concern about the animals' welfares and the necessary of the such efficiency for the validates the safety and the efficiency. Ethical sources of the cells. The sources of the cells in these tissues, engineers raises ethical questions. particularly so considering the use of the embryonical stem cells, the worst adult stem cells. Next slide, please. Regularities approvals. The regulation approval landscapes of the tissue engineering provides the complex as these products often it fails into the multiples of the categories like drugs and the medical devices. Public perceptions. The maintaining the positive public perceptions of the tissue engineering is the essentials for the approvals. Next slide, please. Future properties. Personalized medicine. So the advantages is that the personalized medicines could allow for the development of the treated tissues, the engineering producing that meets the individual patient needs. Regenerative medicines are the integrations. The integrations of the tissue engineering with the regenerative medical the approvals may leads to the more efficient therapies for the particular person. Next slide, please. Bioprinting technologies. The involving 3D printing, 3D bioprintings are enables the printings of the placements of the cells and the materials to create the complex tissue structures that closely resembles the native tissues. Collaborations across the disciplines. The increased collaborations between the engineers and the biologists and the clinics and the regulatory bodies they will facilitate more efficient translations for the bench to bedsides and address the both technical challenges and the ethical considerations. Next slide, please.
Examples: bioprinting. The 3D bioprinting of biological tissues using Bioinks has seen several projects, particularly in the areas of tissue engineering and regenerative medicine. Here are some notable examples. 3D printed skin, Wake Forest Institute. Project. Researchers at Wake Forest Institute are regenerative medicine successfully created a 3D bioprinter that could print skin cells directly onto a wound. The printer uses a scanner to map the wound and then prints multiple layers of cells, including fibroblasts, to promote healing. Impact. This has the potential to revolutionize wound care, especially for burns and chronic wounds. Heart patches. In 2019, researchers from Till University bioprinted a tiny heart using human cells and biological materials. Although the heart was able to a size of cherry and unable to pump blood, it had cells that could contract. Impact. This marked a major step toward bioprinting fully functional human hearts for transplantation, which could address organ donor shortage. Cartilage and bone repair. Bioprinting efforts focused on creating hybrid tissues composed of cartilage and bone, which can potentially be used to repair complex injuries in joints. Impact. These bioprinted tissues could be used in orthopedic surgeries and treatments for conditions like arthritis. Functional blood vessels. German researchers at the Fraunhofer Institute developed a bioprinting technique to produce functional blood vessels. They used a bioink in that combined natural polymers and live cells, which formed a blood vessel structures. Impact. This breakthrough is crucial for engineering larger tissues and organs which need blood supply to survive and function.
Hi, myself Shalu. I'll be explaining about real world application and its outcome. First application is drug testing and development. Application: bioprinted tissues such as liver, skin, and heart tissues are increasingly used in pharmaceuticals research to test new drugs. By printing human tissues, companies can predict how much organs will respond to a drug without relying on animal models. The outcome of drug testing and development: a bioprinting company, Organovo, has developed bioprinted liver tissue that pharmaceutical companies use to assess drug toxicity. This has helped reduce the cost and time involved in drug testing while providing more accurate results. Next slide. The second application is cosmetic testing. Bioprinted skin is being used by cosmetics companies as an ethical alternative to animal testing. This printed tissue can simulate simulate human skin reactions, making them ideal for the safety of skincare and products. Outcome of cosmetic testing: L'Oreal has partnered with Organovo and other bioprinting companies to use bioprinted skin for testing products. This reduces the need for animal testing and allows for more precise safety data. Next slide, please. The third one is wound healing and skin grafting. The application for this is bioprinted skin and tissue patches are used for treating severe wounds, burns, and ulcer. For example, 3D printed skin can be tailored to the patient wound, enhancing the healing process. The outcome of this application, wound healing and skin grafting: the Wake Forest Institute's 3D bioprinter that prints skin cells directly onto wounds is being developed for treating burn victims and other severe injuries. This technology has the potential to accelerate wound healing and reduce scarring. Next slide, please. The fourth application is orthopedic implants, cartilage, and bone repair. The application is bioprinting has made advancements in creating customized bone and cartilage implants to repair injuries or degenerative conditions like arthritis. The outcome for this is ETH Zurich successfully printed hybrid tissues composed of bone and cartilage, which are being tested for their effectiveness in joint repair. This has the potential to improve orthopedic surgeries such as those needed for arthritis or complex bone fractures.
Thank you so much, guys. As we have went across the PPT, traversing different aspects of what is 3D bioprinting. So now we will look one for a video in which it will explain how this process looks into the reality. So be tuned. So now in this video, I hope the video is visible to everyone. In this we can see there are four different types of print heads which are filled with four different types of bioinks, and the cells which are being used for those bioinks are mentioned below. So now this is the fifth biohead, the fifth print head, which is filled with fibrin. Now we can see these bioinks are getting placed or deposited on and later it will get into the formation of a particular organ of which the CAD model has been given to the 3D printer. In this way we can see the precise drop placements is being done by the machine or the print head. Now we have made the first layer of the cells or the bioink placements. Now this is the fibrin which is used for the maturation process. Now this is one of the most important process which helps in the specializations of the functionality of these organs or the particular cells which are being printed. This process will be repeated continuously till whole the CAD model is printed. Now you can see in this video. Now we will see basically how any organ is being formed through the bioprinting. Thank you so much, guys. Through this video we can see how we can print any model which has been used. Now I will request everyone to join the quiz so that you can test your knowledge, as the link is being present in the chat box or you can see on the screen present. So I will request active participation from my fellow mates.
Prasad, can you please send the link on chat?
Yeah, actually I have shared the link itself. You can join there. Uh, have you guys joined? Can I
Yes,
start the quiz?
Priyanka. Yeah.
So the quiz started. So the first question is, what is the 3D printing primarily used for? So you can answer. Yes, it's a right answer. Manufacturing biological tissues and organs. Now the second question is, which of the following material is commonly used as bioink in 3D bioprinting? Yes, it's right. Hydrogels. What are the primary advantages of 3D bioprinting in organ transplantation? Yes, it's the right answer. In 3D bioprinting, what terms refers to the structural support used to maintain tissue shapes during printing? Yeah, that's the right answer. And last question, which type of bioprinting uses light to scaffold bio ink layers by layer? Yes, perfect. That's the right answer. So, guys, as you all stay tuned throughout the PPT and everyone was uh, listened to it, they participated in quiz. Now, I would like to ask if anyone is having any kind of doubt, you can ask here. We will like to explore other topics too.
So I had a question. So about bioinks, how do they maintain the balance like between the printability and the biological compatibility?
The bioink are designed to have a right viscosities for the printing and contains the nutrients and the biomolecules to supplies the cell survivals and the growth. The challenges lies in the ensuring that the ink is both mechanically stable during the printing. I think that's enough for your answer.
I hope you got the answer in a perfect way. Any more doubts?
No, thank you.
Okay, so I hope we have explored through different topics, so different parts of 3D bioprinting, and thank you, sir, for giving us such a beautiful topic. Thank you so much, guys.